So our next speaker is Amanda Buch, who will be talking about cluster-aware machine learning of robust brain behavior associations for precision neuropsychiatry.
Great, thank you. So my name's Amanda. As Elvisha introduced me, I'm excited to be here. I'm a neuroscientist and machine learning engineer at Weill Cornell, and I'm going to tell you about my work on cluster-aware machine learning of robust brain behavior associations. And here's a conceptual illustration I did of this idea. And I don't have disclosures. So the idea here that we're concerned with is that neuropsychiatric disorders are very heterogeneous. For example, a neurodevelopmental disorder, autism, describes a really broad group of people. And in our lab, we're interested in using machine learning to learn from brain measures, like from resting state functional connectivity, and of behavioral measures or clinical metrics, and to see if we can identify these latent brain behavior dimensions or descriptions of a disorder like autism, for example. And then when we have these brain behavior dimensions, can we then learn from these dimensions subtypes that are along the dimensions? So in my previous work, I did this in autism dataset. This was from the Abide dataset. I won't go into this here, but I identified these 4 types of autism along 3 brain behavior dimensions using a 2-step approach, first embedding data and then clustering on it. And this replicated also in out-of-sample data. But an interesting thing we saw is that the brain behavior dimensions we identified, you can see the individuals defined by a subtype, you can see that although they're distinct from each other and replicated, they're kind of close together. And if you look along just one dimension here, you wouldn't be able to see how to separate them into clusters. So this leads to this interesting dilemma or question, which has been a big one in the field. Should we think about disorders as being dimensional or as clusters or subtypes? And so an important thing to note here is that when you apply clustering onto a global embedding space, which people usually do, and sometimes work, there's some limitations. So this actually ignores potential cluster structure that could be in the brain behavior associations. So the idea here was, can we now develop a new methodological approach in using machine learning that could define subtype-specific brain behavior dimensions? And this cluster-aware embedding is how I call it, and we want to jointly and hierarchically reveal these low-rank clusters. And so I'm going to very briefly overview this, but please talk to me at the poster if you want to hear more about all the math or read the paper that's come out. So the idea here simply is that we are designed this framework where we can combine latent factor methods, for example, truncated singular value decomposition, which is like PCA, or canonical correlation analysis with a convex clustering penalty. So you start with the data, as you can see here. The general problem is, can we simultaneously trade off minimizing the embedding error while also minimizing allowing the embeddings to actually cluster and separate from each other, so that lambda there is the convex penalty. And I have 3 algorithms that implement this approach for a cluster-aware PCA, a cluster-aware locally linear embedding, and a cluster-aware CCA, which is a multi-view version. And so this is just a very simple toy example. If you look on the left, you can see ground truth. In B and C, if you first cluster or embed the data, or embed and then cluster the data, you can't recover the clusters. Using our approach, you can recover them perfectly. We get this model path of the solutions, which also gives us a hierarchy or dendrogram of the clusters. So that's cool, that's toy data. What does it look like in real data sets? So I benchmarked our approach against labeled data in 17 real-world data sets across a range of state-of-the-art methods. And you can see that our approach is very competitive or outperforming many other approaches for clustering or clustered embedding in small data sets and also in large data sets. So tens of subjects to thousands of subjects. The variables could be tens of variables to hundreds of thousands. Here's a really simple example. This is more in the paper. This is only 45 individuals. This is COVID-19. You can see in A, if you do this 2-step approach, it can't work, doesn't recover them. Our method recovers them perfectly and finds these distinct associations to this particular protein. And here's an example now in that same autism data set. And what I'm showing you here on the left, this is what I showed you before, looking just along one brain behavior dimension from the previous study. You wouldn't be able to see the delineation of this subtype here, which had a low verbal IQ, but had core symptoms or core behaviors of having a lot of deficits in the two other core areas of autism. But our approach actually can find a very strong separation, as you can see here in this video. And so that really increases our confidence that this is true clusters or subtypes and not just dimensions. And we also learn the dimensions we learn are specific to each subtype. So if you can see on the left here, these are correlations between the behaviors or the resting state functional connectivity with the canonical variant, which I'm calling the P3C8 variant. That's the component for each of the clusters. You can see that they have different associations. So without going into detail more as in the paper, this is really cool and exciting and motivates also looking at this in future data sets. So I'm going to just show one more example here. This is a really widely used data set, which the other speakers have just talked about. It's this adolescent data set from the ABCD dataset consortium. Here's an example where if I apply regularized canonical correlation analysis, or RCCA, and then cluster the data, this is what you would get here of one of the dimensions and how it's finding the clusters. And this is relating resting state functional connectivity to measures from the NIH toolbox, so measures related to cognition. However, if you apply our approach, we actually find that we can find two clusters that have really strong separation. And if you look on the left now, the true colors of how these clusters are in the original two-step embedding space is actually different than what that method was finding. So this is really interesting to think about what the meaning of this is. And I'll also show you just a really quick snapshot. As I mentioned, we find cluster-specific brain behavior associations now, which are also defining those clusters themselves. So this is a preliminary analysis showing that on the left. These are, again, correlations between behavioral scores from the NIH toolbox and the resting state functional connectivity metrics, or those metrics with the P3CA variant, that component. So you can see on the left here, those are the associations from the first cluster that was found with our method, and on the right with the second method. And you can see that they're really different brain behavior associations. On the right here, where you see the box plot, NIH toolbox scores by clusters, those are the original behavioral metrics, and you can see that there are strong differences between the two clusters that were identified, but not in age and also not in sex. So this is still kind of preliminary work here, but this gives you an idea of the potential applications for it, which we're excited to continue developing in this data set and also applying in other contexts. So in summary, we're going to be releasing the code on GitHub, so if you're interested, please sign up there and then we'll send out an email when it's released. Again, these are 3 algorithms, but this is a larger framework of combining this embedding and convex clustering penalty. So we're calling this CARV, or cluster-aware routines, for versatile embedding. And so here's like a very quick summary. We introduced this now cluster-aware embedding strategy, which can be applied in precision neuropsychiatry, but also in other contexts. And along with these 3 fast scalable algorithms that can solve the linear approach, locally linear, and multi-view analyses. And I've also implemented a mini-batch version of the algorithms, which allows them to scale nicely. And also, our approach, very importantly, doesn't actually require you to specify the number of clusters in advance. And instead, it's outputting interpretable per cluster embeddings, which you can see as a dendrogram, as I showed you previously, and then this is the solution path video on the bottom. And then also, as I kind of gave you a glimpse of, our approach also could identify these neurocognitive subclusters in adolescents when this 2-step embedding approach, so first embedding and then clustering on the embedding, didn't work in that case for finding that separation. So this really strongly motivates future applications. And I'd like to acknowledge my collaborators. So all of these collaborators were my collaborators on the previous work in autism, and Connor and Logan are my co-mentors and collaborators on this new method that we developed. And with that, I will take questions.
Thank you for that wonderful talk. So we have a bunch of different questions. So first of all, in terms of these clusters and the approach that you're using, so will the approach that you're using find clusters if there aren't actually any that exist?
Right, that's a very important question. And the answer is no, it should not. We have done a little bit of analyses in synthetic data set, but there's also a lot of interesting theoretical work that could be done there. But the idea is no, it wouldn't find them. You wouldn't see that separation. We also have an approach that can be kind of like a wrapper that can be a fit to that and also determine that quantitatively.
Great, thanks. And in terms of the clusters that it does find, how do you kind of know what they mean in, I guess, like a clinical sense? Like, what is the meaning of each one and how they're separable?
For sure, that's a very interesting, motivating question. So in the case of the autism data, that the way to think about that is then looking at the associations of the brain behavior associations that were specific to each subtype, but then also looking back at other clinical metrics or behavioral data from subjects in that subtype. And for example, by learning different brain behavior associations, that could motivate thinking about personalized neuromodulation therapies that are targeting specific circuits to manipulate or to improve certain symptoms or behaviors. In the case of the adolescent data set, that's kind of different because it's not a clinical population.
Great, thanks. And one more question. How do you know when you have enough data to actually find these clusters?
Right, that's also a great question. I'm not showing it here, but I'm actually working on a paper that I'll be submitting soon that's exploring this exact question, so you can stay tuned for that. But in general, the current way that you would do that is to look at how the brain behavior or other embeddings generalize, and so I've done that in some data sets in the paper, and they seem to generalize. But that would be, you'd kind of have to fit it and see if they're generalizing.
Great, thank you so much. I know there's more questions on the app, but we can't take them right now, but definitely check out Amanda's poster. Thank you so much.